Hi, I'm Dr. Ray, and I'm here to talk to you about weeks 3 through 7 of human embryonic development. Where you left off last class, you had an embryonic disc that was bilayered, and you had this developing embryo that was attached to some developing sacs that Dr. Hill talked to you about, which we'll go over. Then the dorsal side was the amnion, and then you had the umbilical vesicle that was developing on the ventral side of this embryonic disc. And what happens after this bilaminar embryonic disc undergoes further development is it actually turns into a trilaminar embryonic disc that the embryonic tissues are going to all be derived from. So the process in which this occurs is called gastrulation. Gastrulation is when the cells in the embryonic disc are going to change shape, and they're going to rearrange themselves, and they're going to proliferate and move around to make an additional layer in between the two layers that was originally there before you had gastrulation occur. And this event is very important because it is going to anatomically lay out the embryonic body arrangement. And so once the specialized cells developed called the prechordal plate that you can see depicted here, the cells actually become taller, more columnar in shape, then the body's axis is essentially already formed. So this is going to be the head or cranial region of the developing embryo, and it makes the other part of the developing embryo going to develop into the caudal end or the tail end. And then also will help determine whether or not the embryo is going to be on the right side, the tissues on the right side versus the left side of the developing embryo. And so through this process of gastrulation, where cells proliferate, change shape, and move around, we actually have a structure develops that's called the primitive streak. So you can see the primitive streak here on the dorsal side of the embryo, and it's actually going to be a depression that forms because of the epiblastic cells in this region proliferating and moving downward and inward and cranially. It causes a depression on the dorsal aspect of the embryo. And now when you see that depression, if you were to be able to visualize an embryo, you would be able to see that this would in fact be the dorsal side, and you would be able to tell the right and left side from each other. And although we don't talk a lot about gene expression or induction, it is important to know that all of the events and processes that occur in the embryo are dependent upon the cells interacting with each other and the cells expressing certain genes that allow the cells to maturate and develop in certain cell lines. And so the influence that cells have on each other can really not be understated. And if you're reading the textbook, you'll see a lot of paragraphs about the different genes that are responsible for different roles in development. And although you don't need to memorize all those genes, it is important for you to understand the big picture that this process is occurring. And I like this quote that it is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And so essentially this is the time where the embryo is laid out, and a lot of decisions are made, so to say, as far as what is going to happen through the rest of embryonic development. And so the first sign that this event, gastrulation, is occurring, where we have the two layers here that are going to eventually separate when a third layer is formed, is the sign of this primitive streak that occurs here on the dorsal aspect of the embryo. And actually it's a depression because these epiblastic cells are proliferating and moving downward, and they're also moving cranially as well. And they're going to become an additional layer in this developing embryo that is going to be called mesoderm. And so where we had these epiblastic cells and hypoblastic cells, now we're going to have a middle layer called mesoderm, and the outer layer will be called ectoderm, and the innermost layer will become endoderm. So these are the final sort of three layers that will actually eventually maturate into all of the embryonic tissues. And so that's why you see these terms switch on the figures in the book when you read the figures, and it might be confusing to you where this used to be called epiblast. In the image earlier, it's now being called ectoderm, and that's why. It's because we're undergoing gastrulation, we're having addition of mesodermic cells, and you can see that primitive streak, which is really a result of the migrating cells developing on the dorsal side of the embryo. And so we have some anatomic points that are used to describe parts of the embryo, and the primitive, the node being this caudal or cranial part of the primitive streak that you can see here, and the groove is actually the anatomic technical name for the depression as these epiblastic cells proliferate and migrate downward. So these are just anatomic points. You'll see these terms used in figures, and I don't want you to get confused about what they're talking about. They're just telling you a particular region in the primitive streak. And then if you look here, this is a superior view looking downward on the dorsal side of the embryo, and you can see as this caudal end of the embryo proliferates and grows, although the cells do also move cranially, we add length to the caudal aspect of the embryonic disc. Okay, so we have this cranial to caudal movement of cells as it moves downward, and this embryo starts developing further, and that's something that is hard to appreciate from a stagnant picture, but if you can sort of imagine this occurring, and there's also some videos online if you want to see some animations. And so the big picture here that I want you to be aware of is that here we had this bilaminar disc here that was the developing embryo with the umbilical vesicle and the amniotic cavity being on both the ventral and dorsal aspect of this thin embryonic disc. We're going to have proliferation and migration of cells from the epiblast into the layer in between those two layers, and we're going to add this additional layer called mesoderm, and now we're going to call this most superficial layer ectoderm and the most innermost layer endoderm. And so this is all happening during the process of gastrulation, and we are forming a structure that's transient called the primitive streak. And then this view is superior. So you can see all of these views in your textbook, and that's why I try to bring them up and kind of explain them a little bit better, because it looks different according to what view you're looking at. So try to pay attention to anatomic planes when you're reading the text and looking at figures. And I know you're just starting to learn the different types of anatomic planes, but this might help you put everything together when you consider this embryonic flat disc. All right, and so the last point I'm going to say before I ask you a few questions here in this online module is I want to talk a little bit about a mesenchymal cell. So a mesenchymal cell is a special type of cell that's in the developing embryo, and it's referred to as embryonic connective tissue. And so this cell is pluripotent, which means that it can develop into a lot of different cell lines. So if a cell is mesenchymal, what you should think of is it's a lump of clay that can actually turn into lots of different types of cells, and so you don't really know what's about to happen, and it all has to do with the cell induction and the cell gene expression and the type of communication that cells have with each other during this process. And they look very different from other cells. When you're looking at them, you can see these nuclei here that are very prominent, but then it looks like there's a lot of sort of disorganization in the shape, and all of that is very characteristic of a mesenchymal cell. And I want to point this out because mesenchymal cells is actually not synonymous with mesoderm. Those words mean two completely different things. Although a lot of mesoderm also turns into connective tissue, and mesenchymal cells turn into mesoderm sometimes, those words mean something completely different. A mesenchymal cell is a pluripotent cell that can go into a lot of different directions and become lots of different types of cells, but mesoderm is more of a differentiated kind of cell that specifically turns into certain cell lines and doesn't have the amount of potential that a mesenchymal cell has, and therefore it also doesn't look as sort of disorganized and generic as these cells look. So I just wanted to bring that out now because those two terms are really easy to get confused when you're learning embryology, and I wanted to help point that out now.